Hi guys, so we're back now and we're going to this time look at what happens to the product of the Krebs cycle primarily, which is our reduced coenzymes, NADH and FADH. We've been saying that in terms of the journey of energy, the energy started off in the glucose molecule, that got broken down to pyruvate in glycolysis. In the link reaction, the pyruvate got converted to acetyl units, and the energy then was in the acetyl units. Then the acetyl units entered the Krebs cycle. The Krebs cycle broke those acetyl units down to carbon dioxide, in the process generating reduced coenzymes. So the energy then transferred from the acetyl units to the reduced coenzymes, reduced NAD and reduced FAD. And really now we're going to be looking at how that energy that's made its way to the coenzymes, how that energy is harnessed to produce ATP. Okay, so let's begin. So this is just for a bit of context here. I think I'll remove it now, but it's just to remind you that the previous stage was Krebs cycle. Do watch that video if you didn't, so that we can move on and look at this now, what's going to happen next. So Krebs cycle, main products NADH and FADH, as well as carbon dioxide, but the carbon dioxide goes away into the atmosphere eventually. So now let's look at these two. What role do they have to play, and how do we release the energy that's there and convert that into ATP? Okay, so I'm going to get rid of this now. So we're saying that NAD has been reduced and it formed NADH in dehydrogenation steps, and essentially we're saying that the energy is here. So let's look at now how that energy is extracted. So first thing that happens is that this NAD, that this is the matrix, and this is the membrane space, okay, between the inner mitochondrial membrane and the outer mitochondrial membrane. Okay, so this is important for our navigation. All right, so it's important that, so if this is the mitochondria, most dodgy mitochondria I've ever drawn, but where we are is here. All right, where we are. So that's the matrix. This is the cytoplasm. Pyruvate comes in, Krebs cycle happens in the matrix, link reaction, and now the products NADH and FAD are in the matrix, and now this is the matrix, and the NAD and FAD is going to kind of float up here and take part in the next stage. So that membrane right there, that's this inner mitochondrial membrane, and that's why it's so folded, because by folding it, it increases the surface area, and that means we can get more of these important proteins of oxidative phosphorylation and electron transport chain. We can get more of those proteins in that membrane because it's more folded and there's more surface area. Okay, and at the top there we've got our outer mitochondrial membrane, and between them is the intermembrane space, and that's going to be very important. Okay, so let's get rid of that, otherwise it's going to be extremely confusing. So first thing, NADH, it's got hydrogens, and what happens is the NADH is oxidized, okay? So up till now it's being reduced when it's carried the hydrogens, but now it delivers, it gets rid of those hydrogens and it gets reoxidized to NAD. In that process, it gets rid of its protons. Should I just draw two protons instead of writing it? In that process, it gets rid of its protons and it delivers electrons. Should I keep it simple? Let's keep it simple. Let's keep it simple, guys. So 1 proton and 1 electron. The numbers are not important. Protons out, electrons delivered into the electron transport chain. A series of proteins that have prosthetic groups that allow them to accept electrons and donate them to something that's got a kind of a greater pull on those electrons. So NAD delivers its electron here, the proton gets delivered into the matrix here, or as it's separated, it kind of is here. Okay? And importantly, these proteins have a dual functionality. They're electron carriers, yes, but they can also pump protons from here into the intermembrane space, from the matrix into the intermembrane space. But remember we said that NAD has energy. Sorry, NADH, reduced NAD, reduced coenzymes have energy. The energy is in the electrons. And by this protein accepting that electron, it has now got the ability, the energy, to make something happen. And the thing that we're going to make happen is the pumping of protons into the intermembrane space. Okay? Relabel that over here. Okay? And so this electron then kind of passes through these electron carriers. Okay? And as it passes through these electron carriers are pumping protons from the matrix into the intermembrane space. Okay? And this is important here because what we're doing is we're transferring the energy now from the electrons into a proton gradient. Because if we're moving protons from the matrix into the intermembrane space, we're creating a proton gradient. There's going to be many more protons here if this chain keeps working than here. Okay? So the electron continues to move through. Okay? And the pumping continues. Okay? Now, as we have now created this proton gradient, so now, if we're following the story of the energy, glucose, pyruvate, acetyl-CoA, reduced NAD, and now in the proton gradient. It's very, very important that this is appreciated because now that this kind of stored energy in this gradient, now these protons are much more concentrated here than they are here. And they will have a tendency to diffuse back in if they are provided a route to do so. This membrane is incredibly impermeable to protons. But there is one route provided for them to come back through down there, down that proton gradient, and that is through this very important protein, called ATP synthase. Okay, it's a kind of collection of protein subunits that work together pretty much, almost like an engine. Okay, it can rotate. And in that rotation, it synthesizes ATP from ADP. But what gets it to rotate in the first place? The thing that makes it rotate is the movement of these protons. Very nice. The movement of these protons down the proton gradient. Okay? And as these protons, which are so highly concentrated now in that intermembrane space, as they move through, it rotates this unit right here. And that physical movement, as this goes around, the physical movement is an enzymatic action. It causes an enzymatic action And the function of that enzyme is to add a phosphate group to ADP, creating finally our ATP, right? ATP. And as opposed to the other ATPs that we've made previously in the process, this is not a substrate level phosphorylation. This is not simply using the energy from a chemical reaction to phosphorylate ADP. It's using the energy of chemiosmosis, right? So let's just go through that once again. So this proton then ends up here. Okay, so NAD delivered, NAD has got hydrogen. That hydrogen is then split up back into a proton and an electron. The proton ends up in the matrix. The electron is then moving through the electron transport chain. As it moves through the electron carriers, it allows the electron carriers to use the energy of the electron to pump protons into the intermembrane space. This creates a proton gradient. It's both a pH gradient, because of the presence of so much H plus in one place compared to another, but it's also an electrical gradient because we've got so much positive charge there compared to here. So much more positive, much more negative. So for the two reasons, to balance out these two gradients, protons, that's effectively, that's why the proton gradient is so strong, because protons move back down through the ATP synthase to down both a pH gradient as well as an electrical gradient. Okay? So the proton moves through the ATP synthase, and that movement, that proton motive force, drives the ATP synthase to phosphorylate ADP to ATP. Okay? And as you can see now, it really is, without that, the electron doesn't move through the chain. Without the electron moving through the chain, we don't get the protons moving into the intermembrane space. Without that concentration gradient, without that proton gradient, there's no reason for the net movement of protons in this direction. And if we don't get protons moving through the ATP synthase, then there's not going to be the phosphorylation of ADP to ATP. Okay? So that's one important aspect of this. Okay? Secondly, FAD. Now, FAD is similar, but the way FAD works is that it delivers its electron at a later stage. Okay? So FADH also reduced to FAD. Its electron then enters at this stage right there. So in effect, FAD doesn't pump as much, doesn't result in the pumping of as much protons as NAD does. I hope you can see that. NAD results, because NAD adds its electrons earlier in the electron transport chain, it results in more proton pumping, and therefore it contributes to more ATP production. But FAD, because it passes its electron further down the chain, it doesn't cause as much pumping because it misses one of the electron carriers that does that. And therefore its contribution to ATP production is slightly less. Okay? So I hope this is making sense. Now, what happens to these electrons? Okay? This chain will only work if there's something taking away electrons from complex 4. If this electron stays there, then this protein or this electron carrier won't have another electron carrier to pass its electron to. So if an electron is occupying this space will then be occupied by this electron. And if this electron is being occupied here, then an electron will get stuck here. And if an electron is stuck here, then this one can't get rid of its electron. So if we don't have anything removing the electron from complex 4, we're really going to result in the kind of blocking up of this whole electron transport chain. It only works if there's removal of this electron. We remove this electron, it makes the space for the next electron. We remove that electron, it makes the space for the next electron, and so the chain keeps working. Okay, so it's very important then that something accepts this electron from complex 4, because if nothing does, the whole chain stops. We don't get proton pumping, we don't get ATP production, and we don't get ATP for our bodily processes, all our synthesis reactions, neural conduction, muscle contraction, and we will soon die. Okay, so it's very important then that we keep this chain moving, and therefore it's very important that we have something to accept those electrons. And what is it that does accept those electrons is oxygen. Okay, humble oxygen. Oxygen comes along, and I'm going to show it as one oxygen atom, but remember oxygen is it's O2, two atoms of oxygen covalently bound to each other. But for simplicity, I'm just going to call it oxygen. So oxygen comes along, and what it will do is it will combine with one electron, and it can accept 2 electrons, so maybe 2 NADs, or you can imagine that if it would require 2 electrons, and it can readily get 2 protons from this area, because remember when NAD is being depositing its electron here, it also frees up a proton here. So there's a ready supply of protons in this environment. As well as that, they're coming through the ATP synthase, aren't they? So proton plus electron, hydrogen. Proton plus electron, another hydrogen. 2 hydrogens and an oxygen, H2O, water. Beautiful. Okay, so shall we just quickly go accept the electrons that are coming off this, or that are in this complex 4. If we don't, the whole thing gets blocked up. So we need oxygen to come along. It combines with 2 electrons and 2 protons, each proton and electron forming a hydrogen. So we end up with water. And remember the equation for respiration: glucose plus oxygen, carbon dioxide plus water. And there is your water. Okay? So when it says glucose plus oxygen, I hope you can see now it's a gross oversimplification. But yes, we do need oxygen as part of the equation, because without the oxygen coming in to accept the electrons, the whole respiratory process, or aerobic respiration, does not work. Okay, so oxygen comes in, accepts the electrons, and we have this process continuing. Okay, so oxygen acts as essentially an electron acceptor, and that's what you have to understand from that. That it's because of oxygen's presence, it frees up the electron from complex 4, and combining with protons, it can form hydrogen, and the hydrogen can combine with the oxygen to form water. But most importantly, if we keep getting oxygen coming in, we are still able to then remove electrons from complex 4. And as long as we keep doing that, electrons can keep moving along the chain. And as long as electrons can keep moving along the chain, we can keep pumping protons. And as long as we keep pumping protons in there, we'll have a proton gradient. And as long as we've got a proton gradient, we'll get protons trying to move back down that proton gradient through the ATP synthase. And as long as we've got that happening, we'll keep generating ATP. And as long as we keep generating ATP, we'll have enough ATP for our energy-requiring processes in our cells, in our tissues. And as long as we have that, we will be alive. Yeah? Good. And as long as we're alive, we can keep trying to do useful, good things with that life. Right, guys? Yeah? So that's the importance of oxygen. That's why you need to keep breathing, because you need to keep accepting electrons from complex 4. Okay? Right then. Importantly, if this change stops, how does it affect the previous processes? Well, if this change stops for any reason, and some inhibitors are inhibitors of these electron carriers, and they will prevent electrons from moving, and if that happens, we've already seen the consequence on ATP production. But also, if you stop this process, if the electrons back up all the way to this point right here, NADH doesn't have anywhere to deposit its electron and protons. And if NADH doesn't have anywhere to donate its electrons to, because that space is already occupied by an electron, because it doesn't have anywhere to go, then NADH builds up. And if NADH builds up and FADH builds up, we're not making any NAD anymore. We're not regenerating NAD. We're not regenerating FAD. So Krebs cycle doesn't have anywhere to deposit those hydrogens that are coming off. So if hydrogens are not being able to go onto these coenzymes, Krebs cycle will soon stop, because those processes are tied together. Okay? If there's no acceptor for those hydrogens, that reaction won't happen. So if Krebs cycle stops, then we're not accepting acetyl units. And if we're not accepting acetyl units, pyruvate then builds up, link reaction stops. Even the link reaction, it needs coenzyme, doesn't it? So even the link reaction will stop. So that is the reason why only glycolysis is truly anaerobic in a way. Because the link reaction, the Krebs cycle, and the electron transport chain are all dependent on the presence of oxygen for them to work. If oxygen is not there to accept those electrons from complex 4, this all stops. We don't get regeneration of our oxidized coenzymes. Krebs cycle stops. Link reaction stops. Yeah. Okay. So that is the electron transport chain. I think we've said a few important things, and we didn't really write them down. So let me just make a quick note of them here, which is that this is the stage at which our coenzymes are reoxidized. They are reoxidized, and this is important because it's okay to understand that they pass on their electrons, but it's also important to appreciate that those reoxidized coenzymes are then now freed up to go back to the Krebs cycle and collect more hydrogens from the Krebs cycle. That's important. So our coenzymes are reoxidized. We talked about the pumping of protons, and please be careful to not call it active transport. Active transport implies use of ATP. We're not using ATP here to move the protons, we're using the energy from the electrons. Pumping of protons, we mentioned the proton gradient. We're not really going to get very far without mentioning these things, okay? Proton gradient, we mentioned that the proton gradient is actually both an electrical gradient as well as a pH gradient, and that the movement of the protons down that proton gradient is known as a proton motive force, or chemiosmosis. Okay? Chemiosmosis. And the process that couples oxidation and phosphorylation in general, this thing, combined with the phosphorylation of ADP, oxidative phosphorylation. Okay, so these are the kind of key things. Once you get the understanding in your head of what's going on, this is then the next stage, which is how best to describe those things, and these are the kind of things you need to be saying. Okay, I think we'll leave it there. It has been a privilege and an honor to explain this process to you. If you have any questions, do ask me. I'll be very happy to explain. Yep. And I will say good luck, guys. Thank you.